Good morning, dear students. Today I will the polymerase chain reaction PCR. The process consists of three major phases: denaturation, annealing, and extension. During denaturation, DNA is heated to 95 Celsius to disrupt the hydrogen bonds between complementary base pairs of the double-strand molecule. Denaturation of the template into single strands, heating it's between 94 to 96 Celsius for 10 to 20 minutes. Separate the DNA strand using Tag polymerase, consuming about 5 minutes. The second stage, annealing of primers to each strand for new strand synthesis, lowering the temperature to 50 to 65 Celsius for 1 to 2 minutes allows. The third stage, extension of the new DNA strand from the primers at 72 Celsius for 5 to 50 minutes. Tag polymerase fills missing nucleotides in the 3' to 5' direction away from each primer. DNA polymerase works rapidly. Polarization, thousands of base pairs per minute. Amplification. The amplification stage produced millions of copies within about 45 minutes. The amplification product is termed the amplicon. Each cycle lasts from 1 to 3 minutes. Amplification may be damaged or degraded or unable to replicate during PCR due to limited template DNA size, oxidative damage, and or enzymatic breakdown of the sample. Here in this picture we can see the stages or the steps of the PCR polymerase chain reaction. The first stage or the first step, it's about 1 minute at 84 Celsius. The second step, annealing, about 45 seconds at 54 Celsius until 65. And step 3, extension, it have 2 minutes or for 2 minutes at 72 Celsius. We can see these pictures and the pieces of the DNA. The primer will identify the target region and then amplify it by the PCR processes. The primer are used to produce the DNA sequence. The development of the PCR primers will be capable of amplifying the DNA barcode region to identify the organism pieces. There are two types of primers: species-specific primers, and the second one, universal primers. The length of a PCR primer must be sufficient to ensure that it is present in the target DNA. Primer length. The length of primers directly influences the PCR specificity and also influences the annealing temperature of the template. Typically, primer lengths are from 18 or 19 sometime to 24 bases, depending on the genome size. About the nucleotide GC contents, GC, guanine and cytosine, contains affect the sequence's melting temperature, Tm. The optimum GC contents is between 40% and 60%. The Tm, or melting temperature, is about 56 to 62 Celsius when there are 20 bases within 50% GC contents. 3. The 3' end of the primer sequence must be contains GC clumps consisting of 4 bases. Avoid the repeating sequences like T, 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 or avoid to repeat the other sequences G, 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 and also avoid repeating the sequence like TA, 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 or avoid repeating sequences like CG, 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 etc. 5. The forward and reverse primers bind to opposite ends of the DNA strand. The forward it's moved from 5' to 3', and the reverse moved from 3' to 5', respectively. In this table, you can see the component required for the PCR reaction for one sample. Components, and here like master mix is about a 12.5 microliter, PCR graduator or H2O2, it's about 8.5 microliter. Forward primer 1 microliter, their concentration 10 picomole per 1 microliter. The reverse primer also 1 microliter, and their concentration about 10 picomole per 1 microliter. DNA is about 2 microliter, nanogram per microliter, and their total it become 25 microliter. Here, another table, cycling conditions of PCR for amplification of the CO1 gene. It's one of the examples. Initial denaturation, the melting temperature 95 for 10 minutes and for one cycle. Denaturation 95 Celsius, the melting temperature, their time 45 seconds for 35 cycles. Annealing 55 until 60 Celsius, this is the melting temperature, and their time about 45 seconds per 35 cycles. And stage extension 72 Celsius for the melting temperature for 1 minute, and by 35 cycles. Final extension need 72 melting time for 5 minutes and in one cycle. The total time for the PCR protocol for this example, it's about 1 hour and 42.5 minutes. This is the PCR, and we have like video about this. Thank you very much, dear students.